As Peggy said, I'm a computational stem cell researcher. My lab has two arms. The first is we do a lot of mostly transcriptomic data integration to try and understand cell behaviour. We also have a wet lab arm where we take pluripotent stem cells and grow them into cells of the immune system in a laboratory dish. And these two arms are very complementary. I mean, fundamentally, if you're growing a cell entirely outside of the body, it's never seen the body, one of the fundamental questions you have is how similar is it to its in vivo counterpart? And so these computational models really help us understand the conditions needed to grow the right cell types and also allow us to do that benchmarking. Okay. So pluripotent stem cells, for those of you who don't know, are really remarkable cells. They can be made from any one of you. So if I had your consent and the right ethics, I could take a little bit of skin or a bit of blood or even some cells from your urine and then reprogram those cells so that they behaved like cells of a very early embryo. And because they can be made from anyone, it allows us to now have a library of cells that represent our populations in all of their genetic diversity and health diversity. We can use them as therapeutic agents because we can grow these stem cells into other tissues, heart tissue, neural tissue, tissue modelling the back of the eye, and of course, blood cells. They are increasingly used to support diagnostic testing. So in functional genomics, if you have a suspected disease-causing variant in a gene, but you're not sure that that variant is the cause of the disease, you can model that in a pluripotent stem cell in the right tissue. We can do a lot of drug screening in stem cell-derived tissues. So increasingly, these are being touted as a platform for non-animal testing for drug safety. We're also starting to see some really interesting hybrid applications. For example, there's a company just down the road from me, Cortical Labs, which makes computers powered by iPS-derived neural cells, neural corticoids, and it can play exciting things like ping pong. So these hybrid applications tell us that when donors give us consent to using these pluripotent stem cells for one research activity, we need to be thinking about that ethical provenance for future activities which may not be imagined at the time that we took the donor biopsy. Hengi introduced you to some of the platforms that my lab has developed, and they've really been developed out of necessity and a desire to see broader uptake from some of these newer omic technologies by the stem cell community. So I'm going to talk about two aspects of my work today. The first is curation of stem cell data in the Australian Stem Cell Registry. And the second is going to be about some of the atlases that we've built in the Stemformatics platform. All of the code that I've described is available on the Wells Lab Git, and you're very welcome to check that out. So data curation is one of these things that requires a level of understanding of the experimental conditions and the biological conditions that have been set up around those experiments. So we have a very strict curation pipeline bringing data into Stemformatics, and 30% of data in public domain fails our curation status. And I think this is something we need to talk about very openly, because even well peer-reviewed publications may not actually peer review the data and the code itself. There are lots of reasons that data fails. One of the reasons might be that the raw data isn't available. there are laboratories which accession hack, which means that to get an accession to publish their data, they will only put some of the samples into that accession, but leave samples which are relevant to the experiment out. We see a lot of the metadata describing cell types can be very ambiguous. So sometimes it's hard to definitively identify what the cell is that's gone into a particular experiment. We also can see very obvious examples where samples are annotated as one thing, but they map to a different cell type altogether. So there's been some kind of sample swap or annotation swap. Sometimes even the tubes get swapped in the lab. And then we also see issues of data quality where when we look at the data structure, we can see evidence of over-amplification or just poor sequencing. So curating both at the level of the experiment, is the experimental design appropriate, and at the level of the sample metadata, and then at the level of the data itself is really important. And unfortunately, we see lots and lots of bioinformaticians who will only look at the data structure and actually not really look deeply at the metadata associated with these samples. And you can look at our approaches in more detail in these publications. So you might think that this issue of high data failure could be attributed to the quality of the journal. But in fact, what you can see is that some very prestigious journals like Cancer Cell only has a 1 in 3 pass rate for data quality. Nature, which I publish in, it's a great journal, impact factor of 47, has a 30% failure rate of data in our hands. Science is a little bit better, but I will also say that we maybe don't have as many science papers as other papers. Whereas some journals, such as the Stem Cell Research Journal, which is a low impact factor, has a very high pass rate because they really emphasise technical aspects in that journal rather than necessarily emphasising the impact of the findings. And I'll come back to the Stem Cell Research Journal towards the end of my talk when I introduce how we're using large language models for data curation. So when we think about Australian stem cells, one of the problems that we run into very frequently is that Australians are not very inventive about what we call our cell lines. So you get a stem cell made from the donor, and very often you call it control, or you might call it IPS1. And there are lots of cell lines named IPS1 in the public data system. So this word cloud shows you the frequency of names associated with cell lines just from Australian data sets. What you won't see here very frequently are strings like this, MCRI I10A, for example, because this is a registered persistent identifier, which means that this cell line can be identified in a machine-readable way in papers and in the data sets that they're associated with. And unfortunately, this line is an exception rather than the rule. So poor nomenclature can make it really hard to understand if we are looking at multiple data sets where we've got lots of control iPSCs. Are these the same cell line, or are they just different cell lines from different donors that have been named the same thing? We can also see that in the absence of persistent identifiers, that the names of these cell lines change a little bit from publication to publication. So here you can see comparisons between the original publication, which might be within the last 5 years, and then a publication in the last 2 years where they've reused that cell line, and we've verified that through the reference tracking in the subsequent paper. So about half the time the name is similar enough to be unambiguous, but about half the time the name is either completely different or we can't actually track it to the original paper at all. So this is really concerning because it tells us that there's a fundamental problem with cell line provenance in the metadata of these experiments. And that's problematic for two reasons. The first is these are human cell lines, and so there's an ethical provenance associated with what we can do with the data. And the second is that it's an issue of reproducibility, research reproducibility. So what's the solution? Oh, sorry, it gets worse. So if you have a look at not just the name of the line, but other information like the age of the donor, the sex of the donor, the disease status of the donor, then you can see that if you've got a registered cell line, very rarely do you see conflicts in that information. But if you're just relying on the name of the anecdotal name of the lab line, you can often see a high level of conflict with the original paper, or again, we can't actually verify the details at all. So again, this is an issue about reliability. Now, we look at this really closely because we want to integrate data together from different pluripotent stem cell models. So we want to know, are we subsampling the same line over and over again? And we want to know what the disease status or other variables of the donor are, because we want to be able to see the effect of that within our atlases. So we would exclude data sets that are associated with these core lines. Anecdotally, they go into our lab as part of our shit list, which is unfortunately growing. So what we can see is that about 50% of pluripotent stem cells worldwide are only described through publications. They're not captured within a stem cell registry. And most of those pluripotent stem cells are only described in one publication. So finding that cell line requires you to actually find the publication and then reach out and ask the author for that line. Whereas if the line's in a central repository like a bank or a registry, then you can discover that line ahead of reading the paper because it meets the criteria you are interested in. You've got the ethical consent to use it, it's from the right donor demographics, and it's from a donor with an appropriate type of disease. We looked at how many different registries or cell banks are available worldwide, and this is PhD work from Chi Hu. And what you can see is that there are lots of catalogues existing all around the world. Australia has got about 100 lines registered at the time of this study. It's sitting over here. And that's in contrast to other jurisdictions around the world where there are many, many more cell lines available in these public catalogues. Now, we know anecdotally from interviewing Australian researchers that there are probably about 3,000 pluripotent stem cell lines made here in Australia. So the fact that we can only find 100 in the registries tells us that that's really underrepresented. And this is confirmed if we actually go into the registries and look explicitly for Australian lines. We can see that there are only 52 lines which are fully registered within these registries, and about double that number which are partially registered. This is another form of accession hacking. These people have gone to get a persistent identifier, a registered identifier, to overcome a publication hurdle. They haven't then gone back and filled out all of the details about the technical or ethical provenance of that line. So this work has been published, and the bottom line is that we discovered that most Australian researchers don't register their lines because they just didn't understand that this was a thing. They were really on board with the idea of fair data and fair data science, but they didn't actually understand how formalising name and information about a stem cell line might be part of that fair data provenance. And most Australians rely on papers to find information, but there's also a very large collection of Australian lines which are not yet published and may never be. So that's a resource that could be used across Australia, but is a bit invisible at the moment. Now, we also know that internationally, registration of cell lines is really important, not just in the stem cell field, and the International Society for Stem Cell Research has recently released guidelines for research quality stem cell lines, as well as guidelines for clinical quality stem cell lines. And both of these agree that registration of cell lines is a first step in high quality stem cell research. So in response to this, we've now created an Australian stem cell registry, and we've done that because we can see from the use of other registries worldwide that if you have a local registry with local teams talking about the role of registration and educating the stem cell community, people are much more likely to pick it up and register. You can see that these registries record lots of really useful information. So in addition to assigning a persistent identifier, which is unique to that line, it also gives lots of information about where the line was sourced from, what the ethics associated with the line are, whether there have been any genomic modifications, very explicit about what quality controls have been done on the lines and how they grow, and links to lots of external references. So you can see things like, has it been reprogrammed with a vector which will integrate into the genome and therefore impact on any omics experiments, or whether it's been reprogrammed with a non-integrating vector. Really simple things like that. So currently we have 309 cell lines registered in the Australian Stem Cell Registry. We started with 52, so that's a huge uptake over the last 2 years, but it's still only about 10% of the total number of cell lines available in Australia, represents 23 different disease areas and from 31 groups around Australia making stem cell lines, including 4 main facilities in Queensland, Sydney, and Victoria. So I showed you that we've got about 10% of the Australian cell lines captured in our registry. And I also told you that most Australian stem cell lines are described in papers rather than in international registries. So it made sense that we would try and use large language models to read papers describing these cell lines and retrieve that information. So what we did was we set up our protocols using Haystack. We tried 2 very simple chatbots, Claude Sonnet and ChatGPT-5. We verified this against 100 lines that we'd already manually curated. And we stopped using Claude pretty quickly because we found that it truncated a lot of the answers we needed to. It just didn't fill our cell line retrieval out sufficiently. And this is an example of the prompts that we give. So we try and be very explicit about the field that we want the information to go into and with exactly the type of information we want the large language model to provide. There's been a lot of iterating around these prompts, and I'm happy to talk about that with anyone who's interested. So if we take journals which have got a really strong editorial emphasis on cell line provenance, and so information about the cell line is structured really clearly in the methods of those papers, and Stem Cell Research, which is the paper that had a very high pass rate in our stem thematics curation field, is one of these, then you can see that we can extract a lot of information out of those journals with a lot of accuracy. You can see the average score across these fields is 100 or 99%. That being said, not every field is filled out by every paper. So you can see that cytobanding to confirm genomic alterations is actually only done in a very rare number of cases. Very few people do extensive microbiology screening for HIV or Hep B and so on. And that probably just reflects the pool of donors that they've been sampled from and whether someone else has done that testing before it's got to the IPS lab. So that's surprisingly good, and it means that we can rely on those journals to automate pick up very nicely. If we take journals that don't require reporting on cell line provenance, then it becomes a really different matter. We can see that some things are still easily able to be extracted, like the title of the paper, but there are other things that are very hard to pull out, even things like the first author of the paper responsible, or the custodian of the cell line, can be ambiguous. Information about karyotyping can be really missed very frequently, and you can see that even fundamental things like how they are being passaged is only being picked up accurately 22% of the time. So these stats have all been verified by our human curator. So even though this isn't performing fantastically well for non-structured papers, it is still something that we are using as a starting point now for bringing new lines into the registry. So this is our interface. You can drop a PDF of a paper in here, we'll transcribe it, and then you can move through to this curation process. The curation process will allow you to convert the information in the PDF into a series of JSON files, which can then be imported into our registry database. And with the output editor, it will flag where there are entries that are missing, or if the text is different to what it was expecting, based on all of the other entries we've got in the database. So that's been fantastic for moving us forward on that curation process. So just to summarise what I've spoken to you about, information about the cell lines contributing to an experiment is as important as the data files that we get out. And although we've got lots of ways of automating, looking at data structure, often we don't consider just simple things like cell line provenance. And this information is often missing in the metadata fields of omics experiments and may be unreliable in up to 30% of expression studies. I think this statistic is only going to get worse as we move to new technologies like single cell, because there is much less experimental validation of the cells going into those experiments. You're relying on the data itself to tell you what the cells are in those experiments. But in fairness, biologists need help. They need user-friendly platforms to support fair data compliance, because it's often really onerous to upload this information into an expression database or a registry. And we need to normalise analysts asking for this information, as well as the primary data file ask about the experimental data. I showed you how we're using large language models to form part of a solution, always in partnership with a human curator. And we're using that as an internal validation tool as well if we're importing new data. For example, we have data exchange with some of the international registries, and we always use our curation tool, our large language model, to check the validity of information in those files too. That registry isn't launched yet. It will be launched late 2025. And because we've partnered with Phenomics Australia, if you get an IPS line made with one of the Phenomics facilities, the cost of making the line will include registration of the line. So we are confident that we'll have a much better uptake moving forward. So I wanted to spend a little bit of time talking about my favourite cell type, the macrophage, and why I think building these atlases is so important. And it's because, as you can see them moving around this tissue culture dish, they are the housekeepers of the body. They are there to remove dead or dying cells, remove antibody-associated antigen, and in turn, the communication they provide to tissues is really important for a range of different homeostatic physiologies in the healthy body. For example, the microglia in your brain are always pruning the synapses between your neurons to remove excess neurotransmitter. And they actually do it presynaptically as well. So they're instructing the neurons what can go out. In some tissues, the formation of new blood vessels is driven by growth factors which are made by macrophages. They're really important in regulating brown fat to white fat ratios and your body temperature. They clear away dead cells and tissues. They turn over your bones. So they're part of the potassium-calcium homeostasis in circulation. They're critical for iron recycling. They take up the nuclei of dead and dying, they take up the nuclei of newly formed red blood cells, and they help load those hemoglobins with iron. And likewise, when a red blood cell dies, they recycle the iron. And if you lose that ability, then you start seeing ferroptosis of cells. Cook the cells in the liver, detoxify a lot of substances circulating through the liver. And of course, they're very important for immune homeostasis, and they present antigens to T cells to help boost long-term immunity. So with so many different functions, when we're making these cells in a dish, we're really interested in understanding how close are we getting to these different functions within the body. Is it possible to be able to model these functions outside of the body at all? And that's where the computational models that we've been building have become really important. Now, lots of other people are really interested in macrophages as well. And so I'm going to show you very quickly a couple of really beautiful single-cell studies which have preceded our work, which have really tried to build classifications of different macrophage subsets across many different tissues based on kind of core marker profiles. And I'm sure those of you using single-cell studies will be familiar with looking at top-ranked differentially expressed genes between clusters to get these markers. So Sarah Dick, for example, from the Eplum lab in Toronto, identified 3 broad classes of macrophage in many different tissues, conserved across species. It was this TIMP-D4, LIV1, folate receptor 2 class, the TLF class. That's the epicytotic class that you saw phagocytosing up, eating up all of that debris in the cell culture dish. Then you've got the MHC2 high class. These are the immune responsive macrophages which are ready to engage with T cells. And then you have this class, the CCR2 class, and these are the migratory macrophages, the cells that have moved in from bloodstream into tissues to tackle whatever is happening there. So TLF, MHC2 and CCR2. If we go on to another really beautiful atlas of macrophage subsets, this one from Sarah Teichmann's lab at Cambridge. Sarah, who's one of the driving forces behind the Human Cell Atlas, you can see that those markers are not identified. Those classes are not identified. In fact, she's identifying a very large number of different macrophages. She's using different nomenclature, classical monocytes, intermediate macrophages, intestinal macrophages, and so on. And even if you go looking for the markers that Sarah Dick found, they're actually either absent or they're not present in the DE list because the contrast matrix that Sarah has used is different to the contrast matrix that the Teichmann lab has used. So it's really problematic, this transfer of labels from one study to another. Here's another huge atlas. It was published quite a few years ago now from the Junot lab in Singapore. I think Florent Junot is one of the big macrophage fans of the world, is zealot around how wonderful these cells are. And again, you can see he's brought a lot of different data sets together, harmonised them using Seurat to find this set of what he calls the macrophage verse. And again, identifying subtypes of macrophage, which are not identified in either of the previous two studies. And that's because, again, he's got a different contrast matrix here. So all of these cell labels are useful within a study, but they're not transferring between studies. And I think we really want to ask ourselves whether this approach to harmonising data is really working in our favour, because I think what happens is that we start to lose some of the important experimental or technical variability, which might actually help us tease apart more generalisable factors. So the way the cells were harvested can impact the sex of the donor, and so on and so on. Gets harmonised away when we're trying to integrate a lot of data together. So that's just to summarise what I've introduced to you, that even from a relatively simple cell type, the macrophage present in most tissues of the body, it can be hard to find definitive markers to identify these cells. We can over-harmonise data when we bring it together, so we lose informative information because experimental variation tells us something about biology. So we took the opportunity to leverage some really old-fashioned technologies, which bulk RNA and microarray, because these were accompanied by very deep cell phenotyping, so we knew a lot about the cells experimentally before they went into the profiling experiment. And this allowed us to start building some foundation models for projection of new single-cell data sets. I will say I think there's a big gap here for new reference atlases and also new methods that help us identify the exceptions, the exceptional cell types and the exceptional cell states that don't just aim to enumerate what's already been found, but also to help us help us explain why things are different between two data sets beyond just the way they were measured. So the symptomatics approach is very, very simple. What we do is we look at the data sets driven across lots of different platforms. We examine the main components of variation and we filter genes. So we filter genes where platform is the main driver of variation, and we retain genes that seem to be immune to platform effect where biology is driving the main components of variation. Before we do that, we do a really simple rank transform. This rank transform allows ties to have the same rank, so we keep some of the data structure. And if this is a recursive approach, so I'm going to show you, for example, that if we take all of the data within stemthematics using this approach, we can easily separate cell types, stromal cells from blood cells and pluripotent stem cells. If we then take the 840 samples that have been deeply phenotyped from blood, we can do a new recursive filtering. We get a different set of genes which show us biological separation, and we're removing genes which are no longer informative in this. We can take this group and look just at the myeloid cells. So this is 800 cells now. It's, again, another recursive filtering approach. And a subset of these, the dendritic cell atlas, another set of genes here. When we do this, I'll see if I can get this started. Yep, when we do this, we can really clearly see that the cell clustering lines up to the deep phenotyping status. We can get a lot of additional metadata analysis about it. But mostly this is what we do. We project data that's been derived entirely from the laboratory onto this atlas to ask how similar are our synthetic cells to cells that are being isolated from blood. And here I'm showing you a data set from a group which has done transdifferentiation of blood progenitors to dendritic cells. And in their science paper, they show very high correlation of the primary cells and their transformed cells. But the problem is that's the only comparison they do. When we project it onto our atlas, you can see that their primary cells sit down with our dendritic cells, but their transformed cells do not. They're sitting up with progenitors here. And so they aren't getting the level of efficiency of reprogramming that they think they are. And this is important for them to know, because this difference allows us to say what's missing. What else needs to go into these cells? Maybe it's a small molecule, maybe it's a growth factor, maybe it's another transcription factor that allows them to get something which is more similar to the primary cell type. Oh, I'm really sorry for the quality of these images. So we're using this approach to help us identify cells in new tissues too that haven't been centred within the Stemformatics Atlas. So this is a preprint available from Yidi Deng and Brian Inagam, where we've taken multiple data sets sequencing the inner ear of the human cochlea. And in particular, we're interested in, are macrophages part of that developmental process? And if they are, how many subtypes can we see? So what Yidi did was he compared out the unintegrated data. He's just used SRAT integration here for from the different studies which spanned different adult and fetal ages, and identified, here you can see 8 groups of macrophages very clearly, and we can see them with different levels of frequency across the different fetal and adult ages. And when we have a look at the markers which define these, what we can see are hallmarks of a few of the preceding papers. So we can see Sarah Dick's phagocytotic macrophages, for example, here, and they are really enriched in the early embryo macrophages. And we can see the immune surveillance macrophages, which are almost exclusively expressed in the adult ear. And we can see other stages of macrophage biology which are producing, which are not annotated in prior atlases, but seem to have a hallmark of producing growth factors, which are important for a developmental process. When we project this data onto the atlas, we can see something about the provenance of these cells. So, for example, the crosses here are the data that have been projected on from this combined single cell atlas. And you can see that the adult cells are sitting up here in a trajectory that goes from adult hematopoietic stem cells to a circulating monocyte. And these were the HLA high cell types. And then in green and purple here, you can see these are the fetal macrophages, which follow a fetal trajectory from hemangioblast through to microglia, which are known to have fetal origin. And this heat map or density map shows you how those macrophages are sitting across this atlas from adult through to fetal. So I just want to summarise that part of the talk by saying that we are now launching a new atlas of the inner ear. That's being led by my close colleague, Brian Inagam. It has members from around the world. It's very focused, very grounded in the physiology and anatomy of the ear. Definitely an invitation to participate, and will already be reaching out to a couple of you after speaking today to participate as data analysts. I think it's going to be a really exciting process. So if this is your head and your external ears, the part that they're looking at is this incredibly complex cochlea and the cross-section of the cochlea, which has got the hair cells that actually vibrate when you have sound hooking up to the sensory neural system. That atlas will form part of the human. And the final part of my talk, I just wanted to talk about how the human cell atlas is addressing some of these annotation and curation problems. And I'm hoping that this provides you with some resources that are useful, both for finding really well-curated primary cell data sets, but also some platforms for consensus annotation where you can interact with experts in particular biologies worldwide as well. So the goal of the Human Cell Atlas is to create a comprehensive reference atlas for all types and properties of all human cells, and using that reference to understand, diagnose and monitor healthy living and disease. We produced the first draft of the Human Cell Atlas late last year. It consisted of 18 tissues and organs. You can see that the ear isn't yet part of it, which is why I'm really excited that it's going to be driven out of Australia for version 2. And it's really easy to join the Human Cell Atlas, and it's a great community to be part of. You can join really easily on the website. If you can't find that information, shoot me an email and I'll hook you up. It's a grassroots organisation. That means that we try and make sure that anyone entering into this consortium has got an opportunity to contribute at many levels. You can find jobs connected around the world here. You can participate in community discussion groups like disease. There's a Slack, and there are lots of local meetings here as well. You can explore the data sets available from the Human Cell Atlas on the dataset portal. You can see that there are about 500 different experimental projects there at the moment, with almost 1,000 labs contributing. And to be part of the Human Cell Atlas data set, you have a level of curation. So all of the things I've been talking about have been done by some expert data wranglers, and also the data is available. You may need to go through a data access committee if you're wanting the primary bed files, but that's usually just dependent on you getting ethics at your local institution. There are lots of people worldwide involved in the Human Cell Atlas, including some fantastic people here. And it is now moving from its original focus of building a healthy tissue atlas into a disease focus. And I think that makes it really relevant and interesting for many of us. So because of that, I think there are going to be some really strong partnerships with different funding bodies, and UNESCO has also just, in the last Human Cell Atlas meeting, partnered with us to ensure that we have genetic diversity and capability building in countries around the world. So the cell annotation platform is what I wanted to talk to you in my final few moments. You can see it's available at celltype.info. And think about this as the final step in your cell type annotation from a single cell experiment. So you've done your careful curation of which data sets you want. You've done your clustering, you've integrated the data together. Maybe you've used CellTypist or something like that to identify the different cell types. Now you can come into the cell annotation platform and you can engage with biological experts in that area to really ask whether or not these markers tell you something more about that cluster. So maybe you've identified my favourite cell type, the macrophage, but there may be additional markers in that cluster group that tell you something about where that macrophage has come from in terms of tissue, or what it's doing in terms of its functional role or disease status. And the nice thing about this annotation platform is that it's really community based, so that you can have disagreements, you can have consensus, and you can choose who you want to believe and who you don't. It's a really respectful environment, but you can have really clear evidence for or against inclusion of markers for a particular cell type. Now, this means that for any new atlas coming out of the Human Cell Atlas, you will have this coordinated data ingestion and this coordinated annotation platform. And certainly the new atlas of the human ear will take advantage of this. So, you know, I was looking for a summary slide. Why has my talk been almost entirely on metadata and not data? And it's really important because it tells you how the original data was measured. It tells you something about the biological material, and this information is fundamental, particularly as we move to bigger and bigger models with deep learning. Having high quality data coming in is really important. And it helps us start to agree on consistent transferable cell type labels, which allow common understanding across the community and aren't just, well, it said so in this reference versus this paper. I think this is incredibly important as we move towards building these really large harmonised data sets. So I'll leave it at that for the Human Cell Atlas. You can explore lots of these metadata tiers within the data portals. You can, when you're uploading information, you'll get really clear instructions about what's missing. So disease ontologies, for example, are really important. You can quickly look across lots of different atlases for compliance with these kind of metadata terms. And that allows us to really build scalability. So I would really encourage you to be part of this exciting international consortium. And again, if you've got any questions about it, I'm really happy to answer that. So that really just brings me to a final acknowledgement slide. And I guess the first person I want to point out here is Bryony, who's leading the ear atlas. Chichi, who did all of the hard work with the Australian stem cell community to understand why we were doing so badly at naming our cell lines and helping to build community-led solutions to that. Yudi, whose work on identifying macrophages in public inner ear datasets I showed you, and then Susie here at the back, who is our main curator, both within Stemformatics and within the new Australian Stem Cell Registry, and the registry which is being built in partnership with the Melbourne Data Analytics Platform, and particular thank you to Noel for that. Thanks very much.